environmental and waste management, development of absorbent materials, as well as production engineering, production planning, quality control, work study, and also productivity management. For today, we are honored to have her share her knowledge and expertise with the topic General Concept in Quality Control, Organization for Standards, Conditioning, and Sampling. Dr. Zora, how are you today?
Thank you. Very good. I'm really thankful.
Okay, nice to hear it. Dr. Zora, are you ready to do the presentations?
Yes, I'm ready.
Okay. So, Dr. Zora, do you have the presentation to share on your own, or do you need our committee to share it?
I will share my slide.
Okay, nice to hear it. So, Dr. Zora, time is yours.
Thank you, moderator. Very good morning to all of you.
Itu
Good evening
tantangannya
or
ini
good
tadi
afternoon.
apa prosesnya tadi
Welcome.
pagi?
I am very thankful to the
Enggak, kalau sekarang ini apa?
International Summer Course 2026 at University of Gajah Mada, Indonesia. And they have given me opportunity to be a part of
Penghilangan
this
kulit ya?
course. I am really thankful.
Sorry.
Now I am going to share my slide.
Jadi yang pertama apa tadi, Iris?
Tadi itu cara nih, kan ini kemarin
Direndam.
ada kunjungan bulu sama kapurnya,
Ya.
itu yang dibersihkan tadi.
Oh, dibersihkan lewat ini,
Ya,
drum.
putar, baru diisi air, baru diputar lagi,
Diisi air.
dibuang lagi airnya, diganti lagi.
Ganti lagi ya. Oh, belum dimasukkan apa-apa.
Sekarang ini baru berdiri.
Oh,
Tahapan jadi ini.
oh berarti lebih ke penghilangan kembali bulu ya. Lanjut.
Yes, we can see.
Ini kayak
Thank you.
pengurangan sisa kapur
So,
dari hasil pengapuran kemarin.
is it full screen?
Ini berapa lama
Madam,
tadi
could you
dari
please,
jam 8 ini?
Just press the slide presentation mode. Yes, little lower, madam. Little lower.
But
ম্যাডাম ওই যে নিচে আরেকটু নিচে যান, ওই যে স্লাইড একটা কাপের মতো যেটা চিহ্ন দেয়া আছে।
Jadi berapa lama ini?
একটু দেখা যাচ্ছে না।
আপনার ওখান থেকে দেখেন ম্যাডাম, ওই ইয়াটা মনে হয় ঢেকে গেছে আপনার স্ক্রিন।
2 jam ya?
হয়ে গেছে, হ্যাঁ।
আপনি ওইটা একটু ড্র্যাগ করে সরাই দেন ম্যাডাম, ডানে বামে সরাই দিয়ে
Ya.
ওখানে জাস্ট একটু ওই যে নিচে দেখেন একটা কাপের
Tadi
ক্লিক করেন, অথবা আপনার এটা
Ini garam ya?
হ্যাঁ এই
Kiri.
একটু জাস্ট করেন। হ্যাঁ, থ্যাংক ইউ।
Amonium sulfat. Dikira amonium sulfat.
Is it full screen now?
Dikira amonium sulfat ya?
it's good.
Bukan ya?
Ini yang sulfat.
Oh,
So, okay, today's our topic, general concept in quality control, organization for standard, conditioning, and sampling. So, if I ask what is quality and what is quality control, then all
amonium. Oh, tunggu.
that
Oh, enzim ini.
is the fitness for use.
Oh, enzim ini.
For the purpose. That means
Amonium sulfat yang ini ya.
whatever customer requirement, desire, that fulfillment of that requirement
Ini apa?
or customer demand.
Oh,
that
disulfit.
is quality.
Disulfat mana ya?
Whatever demand, whatever
Sorry.
black and white document the customer is provide, that achievement of that requirement, that is quality. So, if any customer
Natrium disulfit ini kan?
I may give an
Oh, di dalam ya.
that
Tulisannya itu natrium metabisulfit.
that like tearsins, that tear is the quality of that particular product. So whatever demand a customer is asking, that achievement of that particular property, that is quality. And to provide quality activities used to do a manufacturer or service provider, that activities is commonly known as quality control. quality control is a huge thing, lots of things. That means from starting to finishing, a lots of precaution, lots of activities generally used to do. That is quality control. So, whatever specification we develop to maintain quality, that assessment of that particular activities,
Lihat dulu,
like
lihat dulu
initially
dalamnya.
whatever we have, we have to measure the actual activities and we will compare it with that specification. If there is any deviation from the specification to the actual activities, we have to mitigate that deviation, or we have to reduce the effects to achieve the quality.
Kemarin
So
nonton
quality
pesta
control
babi
is
anak-anak
many
Awin
activities,
Alaudin.
and
Langsung jam dari
service
tidur jam berapa?
provider
Iya,
or
oh
end
tidur jam
user
8.30 Pak.
always firefight with this source of activities to
Iya,
maintain
rame.
quality. So is it understandable to all? If anyone responds, then it will be better for me.
Yes, I can understand. Thank you, madam.
Welcome. So here quality refers to the degree to which a product or service process meets to or exceed customer expectation or requirement. It serves as a measure of excellence, fitness, purpose, and reliability, representing that distinctive characteristics that make something desirable. That means whatever customer asking, we are providing that quality. That is standard quality. A systematic process of ensuring products conformation to defined specification, and this process is known as quality control. So theoretically, according to, we can say, quality master Joseph Juran, the measurement of the actual performance,
Ya apa ini?
compare it with standard.
Gani?
If there is any
Gani?
variation or deviation, act on that variation or difference. That is quality control. According to Joseph Juran, we can say this. So, what type of quality control generally industry people or service provider they do? Almost 5 types of quality control are there. One is preventive quality control, another one detective quality control, statistical quality control, on-site and off-site quality control. First of all, preventive quality control. What is that? Preventive. That means we have to take few measures so that no defect will arise, no problem will arise. Initially, we take a few preventive measures to maintain the quality. So
Tadi
no
dimasukkan
defect
amonium
period
bisulfat
or
kah?
no occurrence will happen, so that product quality will
Ini
decrease.
sekarang apa
That
masuk
is
dimasukkan apa?
preventive quality control. Generally, this source of activities is done
Oh, amonium
by the
bisulfat tadi
companies.
mana?
They take
Mana amonium
few
bisulfat?
actions so that no defects or non-conformity will
Amonium.
arise. That is preventive quality control. Detective
Amonium.
quality control, that means once a defect is arised, that you have
Mana
to
nih?
find out the root cause, why this defect has come. We have to find out the root cause. So we will eliminate that root cause so that
Ya. Oh,
recurrence
ini.
will not be happened. Once occurrence is there, we have to find out the root cause and eliminate the root cause so that recurrence will not be there, and we can control the quality easily. And there is another control, that is correction. That means repeatedly occurrence will happen and we will repeatedly rectify that defect. or non-conformity, but that is not good. And according to me, I think it is costly as well, because a lots of material loss will be there, time waste will be there to rectify the situation or to rectify the defect. So the correction, it is not so good. Detective quality control is very good. Once one occurrence will there, non-conformity there, we will find out the root cause, to eliminate the root cause, and recurrence will not be there. So it is quite okay. But preventive quality control, it is the best solution to prevent the defects or non-conformity. And another one, statistical quality control. What is that? Statistical quality control, it is the application of the statistics methods to monitor or to control and to improve the manufacturing or various process, services and process, to minimize the process variation. That means if we can't find the how much or which type of percentage it is there, is it 5% defect, 10% defect, or 2%? If we can't recognize the situation, we can't improve the situation. So statistical control, by this we can control, we can identify the situation of the present, and we can overcome this sorts of problem. And on-site quality control, that is the actual quality control, that means in the shop floor or in the production. Everyone is tangible activities are there, that controlling each and everyone is controlling, will control the quality. That means in the production system, everyone is customer. First, worker or team member, he or she will control, she or she will act accurately, and he or she will pass next person. So next person will be the customer. That means on site quality control, it is the best quality control. Each and everyone will control the situation. And offsite quality control, it is generally done by the top management, and that is not tangible work they do, they will do intangible work. That means behind the scenario, they will take necessary action to improve the situation. So this type of quality control generally industry people do, that is preventive quality control, detective quality control, statistical quality control, on-site and off-site quality control. So to control the situation, what type of methodologies or activities or tools industry people apply? First of all, very popular method, I think everyone will know, that is TQM, Six Sigma, lean manufacturing. Apart from this, there are lots of methodologies, tools, generally industry people applied, service provider will apply to improve the situation. Since like Ishikawa fishbone diagram, that is very popular method to all, and I think we know each and every one that Ishikawa fishbone diagram, or we can say cause and effect diagram, that is also known as cause and effect diagram. That is also very popular methodology. So TQM, what is TQM? Total quality management, that involves every employee. That means whatever, just previous slide I have told, that on-site quality control, each and every one will engage to improve the situation. So every employee in continuous improvement of processes or and services. That means always try to improve the situation.
Mana lembaran
And quality is and continuous improvement, it is endless basically. Whatever the best situation is there, we can improve that situation as well. So each and every one will engage to improve the situation.
Oh
that is
iya.
total quality management very shortly. And Six Sigma, it is a data-driven approach aiming for near perfection, 3.4 defects per million, that is Six Sigma. But in our footwear industry, leather products industry, or leather industry, it is very tough to gain or achieve 3.4 defects per million or Six Sigma. If we achieve 3 Sigma, 4 Sigma, that is over plus for us. As footwear industry, leather products industry, or leather industry, it is highly labor-oriented industry. So it is very tough as a human being involved for the activities. So it is too tough to achieve Six Sigma. But still, we have to try for the betterment of the situation. So for Six Sigma, what we use DMAIC framework. What is DMAIC framework? DMAIC, that means D for define, measure, then analyze the situation. If we can analyze the situation, we will be able to improve the situation, and finally to control. Or we have to control the situation, we have to maintain the
Dek,
situation.
RAM 1M,
That's
Dek.
Six Sigma approach
RAM running, Dek.
framework.
Ini saja.
Again, I am telling define, measure, then analyze and improve, and control. We have to maintain the situation to reach that quality level, acceptable quality level. That's Six Sigma. And lean. Lean, what is lean? Lean and lean, we can say, focuses on eliminating waste and maximizing the value addition activities. That means we have to maximize the profit. And if we able to eliminate the waste, then that is possible to use maximum use of our resources. Otherwise, resources will be waste. That may be related to material, maybe man hour, maybe other overhead materials also. So our main target for lean tool, that is eliminating of waste, so that we can improve our situation, we can gain the maximum profit, maximum quality. And with lean manufacturing nowadays very popular the whole world, to minimize waste and to utilize our resources in better in very proper way. That is lean manufacturing. There is another very interesting, that is PDCA, or effective tool, that is PDCA cycle. Though I didn't show that tool, that is PDCA: plan, do, check, and act. So quality control versus quality assurance. Quality control, that means in the process or product oriented activities we are doing here. But quality assurance, it is really process oriented activities. That means we have to develop, we have to make a standard SOP. So that SOP means standard operating procedure. So we can, by the standard operating procedure, we can do our
Di paket timbangan
activities
digital kan tadi,
properly,
Dek?
and
Untuk penimbangan.
non-conformity activities will be arise due to the standard
Oh,
operating procedure.
oke oke. Kira
Staff training program, we can
timbangan
improve
digital yang
our
itu.
staffs, we can provide the staffs, provide training to the staff, so that their skill will be improved. International quality audits, international person expert will audit the situation. They may show the difference, whatever standard is there, we have to act on that. So quality assurance, that means we are providing quality with
Deliming itu
confidence.
apa lagi?
Once
Deliming itu apa
we
lagi?
can assure that yes, we are doing activity
Oh,
with
oh iya iya.
standard, so
Maksud
our
maaf
product
maaf, Dek. Yang
will
jelas
give
dari
the
kita
maximum quality,
ya
maximum
begitulah.
will maintain
Yang
the
satunya
maximum
pakai Bayu untuk itu
standard or
UGM.
whatever specification is there, customer demand is there, we will be able to fulfill through this sorts of activities. So quality
Iya,
assurance
cari yang belum ada.
one provider, service provider, or
Hah? Iya, baru mulai sekarang ini.
industry people, they will say that we are
Jalan.
100%
Jempol-jempol itu.
we are able to provide 100% quality. That means we are providing quality with confidence. We are confident enough this product will be satisfactory customers, and that is how people make the warranty guarantee. Because all sorts of raw materials, whatever industry people use, they ensure the quality of all sorts of raw materials. They ensuring the quality of standard operating procedure, so that they can achieve our final target for the customer. That is what it is. And quality control, it is totally product oriented. It consists of raw materials, original product testing, and laboratory analysis is there to ensure the quality of all sorts of raw materials, so that we can achieve our targeted product in best way. That is quality control. So there is little bit difference with quality control and quality assurance. So now we are discussing that we have to ensure the quality
Apa yang
of
dimasukkan lagi
all
nih?
sorts of raw materials
Oke,
before
oke.
mass production. So how we will ensure the quality? We have to follow some international standard. That means internationally recognized standard. We have to use this standard to ensure the quality of the raw materials. So what sorts of international standard we are using in country like Bangladesh or Indonesia or India, or whole world? That is ISO method. I think we all know what is ISO. That is International Organization for Standardization. That is ISO. International Organization of Standardization, they develop for all types of material, products, service. They have developed lots of standard for the whole world. That is ISO. And then IEC, that is International Electrotechnical Commission, they are providing some method standards for the electrical and electronics and related materials or related products. Then ITU, International Telecommunication Union, they are providing service or providing methods for radio activities or radio, like this sorts of product. And for leather or allied materials, there is one society, that is International Union of Leather Technologists and Chemists Societies, IULTCS. That is very popular for leather testing or footwear or leather products. That means leather oriented materials and allied materials. We are using these methods, standard method, that means world recognized methods, that is IULTCS. So there are three commissions is there for IUC for chemical testing, IUF for fastness of dye or dyed leather, and IUP for physical testing of leather. So there is three commissions. There are three commissions in IULTCS. They are maintaining the quality. They are providing the test method for leather and allied materials. So again, I am repeating: IUC for chemical test, IUF for fastness, and IUP for physical testing of leather. So apart from international standards, there are some other standards, regional standard or national standard. There are hundreds of standards, regional standards are there. I am mentioning few popular, that is EN, European norm. They are providing mostly safety methods. European are very popular. Even I am also using these methods to provide the test. And then ERA organizing for standardization, ERSO, EASC, Euro Asian Council for Standardization, Metrology and Certification. There is another regional standard, very popular, ACCSQ, that is Asian Consultative Committee for Standards and Qualities. PASC, that means Specific Area Standards and Congress. So these are the regional standard. They are providing also lots of standard SOPs to provide the quality. And national standard, very popular. In our institute, Institute of Leather Engineering Technology, University of Dhaka, Bangladesh, we are also using this standard, national standard method, that is SATRA, very popular. Shoe and Allied Trade Research Association, UK. We are using this method. DIN also very popular. Das Institute for Norman, that is German method, German Institute for Standardizing. They are also providing lots of methods, standard methods.
Tadi
And
setelah direndamkan semalaman tidak dibuka itu.
so there are
Tidak dibuka anunya,
regional
drumnya.
standard,
Langsung.
national standard, various of standard
Ya.
methods are there. By using
Langsung
these
putar ya.
standard methods, we are maintaining our quality, all sorts of raw materials related to leather,
Ya.
related to footwear materials, and
Bagi di dalamnya bagaimana perubahannya?
assessing the quality before mass production.
Oh, tidak ada bulunya ya. Warna apa? Berwarna
these
apa
are the
tadi?
challenges, quality challenges related to like
Coklat, masih coklat ya.
visual defects,
Tapi bulunya udah nggak ada ya.
chemical safety. These sorts of challenges are there to maintain the quality. If we say visual defects, that means color inconsistency or unevenness across the surface, or it is very difficult to manage the surface. That means natural grain pattern. It is very difficult for leather, but still it is unparalleled leather for leather to same quality by other synthetic materials, but leather is unparalleled due to the few properties like comfortness and elegant look. So still, during manufacturing, it is very difficult to manage balance the quality, or to balance the color evenness, then grain pattern, like this sorts of thing. And structural failure, if tanning process is not good, then maybe poor tensile strength or tear resistance may occur due to the improper tanning processes. And chemical safety, obviously, it is very essential to ensure the compliance with restricted substance such as chromium 6, azidize, then formaldehyde, etc. So to ensure this source of quality, what we do? We have to assess in two ways, like qualitative analysis or quantitative analysis, quality test or quantitative test. So what is qualitative test? We can test quality by without any instrument. Like lots of industry in the whole world, maybe they don't have the in-house laboratory. Like they don't have in-house laboratory. So what to do? They have to ensure the quality before mass production. So they do few activities to ensure, to anticipate the quality. So that is qualitative test. That means this source of test will not give you the quantitative value. That means this source of test just to anticipate, you can anticipate the quality by carrying out. That is known as, in our sector, thumb test. that is very popular. Leather engineer or leather technology, they always carry out this sorts of test. That is thumb test. So if I am asking what is thumb test, thumb test is thumb test is source of code.
Iya, iya. Maaf,
Leather engineer
dapat
or procedure
nggak ada
they use
penumpang
to
nih.
assess
Ini
quality
tipis,
by
nggak jadi
pulling,
tipis ini
stretching,
HP sih.
and then
Ini
rolling
baik
and
deh,
folding the
tipis
leather with
tipis
their
sekali.
hands. So they can
Minta maaf
assume
ya.
some quality.
Oh, ada semutnya.
Even in other body organ like eyes, nose, ear, skin, by using this source of organ, we can also assess, we can assume the quality. So these sorts of tests are known as non-destructive or thumb tests. If I am giving, I can give example like looseness. We can assess looseness. Here you can see one pictorial image. They are assessing the looseness of the and roundness of the leather. So we can assess looseness, roundness, then run property as well. That means this leather will elongate or not. We can feel it. We can is the surface is the leather is smooth enough. We can see. We can identify the fiber strength, leather to leather strength without any instrument. Right? Yeah. We can without any instrument we can assess it. But
Disapu.
here the disadvantage is we will not be able to get any data from this source of test. Just I am repeating, we are anticipating the quality without using any source of instrument or in any source of machines or equipments. But why these are so popular? Because in all industry they don't have lab. So physical testing laboratory. So they used to do these sorts of tests. So color fastness, flexibility, we can assess it by carrying out spacing, rolling, folding the leathers. So for a laboratory test, there are a few types. Non-destructive
Tadi sudah ditimbang kah?
test. Again, we can
Sudah
classify
ditimbang?
into static and dynamic test. Static or
7,5.
dynamic test for destructive, for non-destructive test. So if we go for laboratory test, then we need a few machineries, environment, a skilled person, then instrument. But instrument, it should be calibrated. So and we have to maintain environment as well. That is known as conditioning. So laboratory test, we here basically we need for lab test. First of all, we need SOP. What is SOP? Earlier I shared SOP is standard operating procedure. That means we need standard, international standard, regional standard, or national standard that is recognized in the worldwide. We need this SOP. So without SOP, we can't carry out the lab test. It is very essential to provide test report or to analyze the leather quality, inherent quality. So SOP, it is number one to carry out the laboratory test SOPs. That should be international standard or regional standard or national standard, but it is worldwide recognized. It should be worldwide recognized. I'm telling the SATRA, it's very popular method. DIN, it is very popular method. DIN. So SOP first of all. Then we need sample. That means you have to collect accurate sample from the leather. So this sample, if we are not able to collect sample properly, then our test report will be maybe it will not be accurate. So sample location or collection of the sample, it is also very important. Then instrument, we need instrument that must be calibrated. That means we are carrying out test. Maybe I can give example: heat fastness test or heat resistance test for leather. So we are starting the test from 100 degrees Celsius. Is it 100 degrees Celsius? Is that machine is providing 100 degrees Celsius? We have to ensure. That is calibration. That means we have to calibrate the machines so that load, temperature, pressure will be accurate. So machine calibration, that is also very vital to provide the laboratory test. Then the skilled person, people who know, obviously they will maintain the they will carry out the test. And last one, that is environment, that is conditioning. That means proper temperature, humidity must be controlled for the lab test.
Sorry to interrupt you, Dr. Zohra. I just
Yes.
want to remind you
Yeah.
that you have 10 minutes remaining.
Okay. Thank you for your information. So, sample size, I am telling just few second earlier. That is sample size and sample location or sample collection is very vital. So, how many pieces or which pieces should be collected from a lot? Suppose we have a lot, there may be 301 to 500 pieces is leather are there. So according to our subcontinental process, like country like India, Bangladesh, or Pakistan, Sri Lanka, we are using this statistical monumental data according to SS Dotto, very popular to all of us. That is, I am showing the, if the lot
Gol
size
lah ya.
is
Eh,
501
apa?
and above, in that
Berapa?
case, we have to collect 20 pieces maximum. So,
Tipu dulu, Isna. Isna,
um,
tip.
N by N piece should be collected. That means for visual assessment or for thumb test, we have to collect, if the lot size is 500 or
Kenapa menggunakan nih
301 to
ordernya?
500 pieces, in that case, we have to collect 15 pieces for the visual observation. And how we will collect? It is N by N piece. That means lot size number divided by collected sample, and that is the number that we have to give the variation to collect the sample. And here I have shown one example, small example. If there is a lot containing 365 pieces of leather, in that case, how many pieces and which pieces should be collected? 365, it is in between, according to previous slide, it is in between 301 to 500 pieces. So we have to collect 15 pieces, and the gap between the two pieces we are collecting, that is 20. Very simple calculation is there. But it is totally depend on opinion of the user. To collect the sample from the lot, it will totally depend on him or her, or he or she can randomly collect the sample from a lot. Depends on in its inspector. But this source monumental data we are using
Oke
in
sih.
our subcontinent to collect the sample from a lot. For laboratory test, there is also one monumental data, right, like previous one. If the sample size, lot size is 601 and above, in that case, we are taking the sample maximum 5 for laboratory test. And again, N by N piece, or according to the sample collector or inspector, he or she may collect from anyone. But number of pieces, it could be 5 for laboratory test. So environment, it is the one of the important factor to carry out the laboratory test, that is standard conditioning. As we know, the climate of the whole world, it is different country to country, region to region. So leather, it is fibrous materials. So depend on moisture or humidity or temperature, inherent quality, inherent property of the fiber, natural fiber, will change. That is why we need to standard the
Masih
environment
dipakai, Kak?
of the laboratory
Ya,
so that
tinggal anunya, tinggal
even
praktek.
there we will be
Tinggal
able
lagi sudah
to
basah.
gain more or less similar property, similar value
Oh, yang
from the
tulisannya
data.
jelas deh.
So what we
Pasukannya ya.
do according to
Kata jelas
ISO
ini media.
291 and ISO
Bisa juga
534,
terbaca.
IUP
jelas
3
sekali tulisannya.
Dan
considering
bisa juga.
material, it is
Bayu saja
obtaining
tulisannya juga jelek
23
tapi
degrees
apa
Celsius
dianggap
plus
jelek ya
minus
kan?
2 degrees Celsius with relative humidity 50% plus minus 5% for non-tropical country. And if it's tropical country like Bangladesh, 27 degrees Celsius plus minus 2 with the relative humidity is 65% plus minus 5% relative humidity is maintained, and total time is 24 hours for conditioning the leather sample. Apart from this, earlier it was classical conditioning. It is 20 plus
Banyak deh.
minus 2 or 21 plus minus
Oke,
4
Kak.
degrees Celsius
Begini,
and
Dik.
65 plus minus 2% relative humidity
Apa lagunya,
for
Dek?
maintaining for condition the
Kita
sample
udah dimasukkan
before
belum?
main
Oke.
test. So before main test, we have to have condition the leather sample to achieve the standard result or accurate result from the test. So how could we collect the sample from a
Maaf deh ya,
particular
Bayu
if
ingin
the sample
charger
is full leather or
mencet
side,
dulu.
then how we will
Tahu
collect
ini
sample from this
cari
portion?
dulu charger ya.
I am showing
Ini
here that
ininya Kak Ilham
two
ini.
pieces of
Ini dulu.
leather. In that case, we have to make a center line in the backbone. That means here we can see C and B line. It is the center line of the leather. From this portion, from here, we have to divide equally in 3 points, like A, B, then A1 and AC. So from this point A, we have to draw one particular line from the center line. And we can see clearly HKJG or GJ SK. This is the square area from where we have to collect the sample for physical test, because this portion is the best portion of the leather. So if we collect best portion, then we'll be able to achieve the best result for the leather. And this test report will represent the whole quality of the whole leather. So we have to collect. But here one you can see AE is the 50 millimeter. That means we have to avoid 50 millimeter from the center line. Why? Because if we collect from this area, it will show the dubious property. Why? Because this is the backbone region of the sample, and this is the location for part. Again, it is almost similar, and we can summarize our session. That is quality. We have discussed quality and quality control, national and international standard, collection of sample from a lot, and collection of sample and conditioning of the sample position. So should I stop here, moderator?
Okay.
Then thank
Thank
you.
you very much for Dr. Zohra for your insightful material, and it's been helping us to create basic understanding in general quality control in leather industry. So now for the discussion, is there any participant that would like to ask a question for offline or online participants? Is there anyone? Okay, then maybe I will ask you a question, Dr.
Yes.
Fatima. Maybe based on your experience, What is the common mistakes found in sampling and laboratory testing procedure based on the general quality control?
Okay, thank you for raising the question. Generally, industry people, they are giving the sample. Country like Bangladesh, I am sharing this experience. Industry people give us the sample, and immediately they are asking for give me the test report immediately. So, condition it is very tough. It is very tough, uh, and another problem is um, taking the sample. They are not taking sample, or people are not able to taking sample from the exact location, because industry people, they are providing sample maybe at a square shape sample. They are not just providing the exact location for the sample, so it is very tough to achieve the quality portion, to take the sample from that. This sorts of problem may arise. And another is machine calibration. If machine is not proper, then it is very tough to get that accurate result, or exact result from the test. So these sorts of problem may could be happened.
Okay, well, thank you. It's very, I understand very good for your answer. So now we got more question in the chat box. I will read it for you from Sangram Wandekar. He asked about what is your opinion and perspective on plant-based or vegan leather?
Vegan leather. Yes, vegan leather nowadays, it is popular. Uh, uh, as you know, resources limited in this world. So if we use material from the nature to develop the leather, vegan leather, or so it is, I think it is helpful for us, for environment as well. So leather processing, it is highly polluted industry. So if we go for that, that may help to reduce the environment pollution. And nowadays, people do research on vegan leather, and they develop very good quality vegan leather. So I think it is positive. It is positive for the environment to develop vegan leather. It's positive for me also.
Okay, thank you, Dr. Zubra.
Thank you, doctor. So can I get a chance to interact with her?
Yes.
Just a minute, ma'am. I will first of all I will show you some samples. Actually, I am working on this vegan leather concept.
Okay.
Yeah, I am from Indian Council of Agriculture Research, Central Citrus Research Institute,
Okay.
and now currently we are developing some materials, bio-based materials. Is my video is visible?
It is visible.
Yeah. Also, this sample like this we have prepared. You can
Okay.
see.
What is the thickness of this vegan leather?
It's 1.5 mm.
1.5 mm. Okay.
Yeah, and
Looking good.
we are
Looking
also
good.
designing some samples like these pouches. It's for
Oh,
mobile
okay.
and small purse.
Understood.
Ladies can use, and it is very easily handleable. So, ma'am, just I also want to ask one question.
Yes.
We are also facing some problems like so these materials are not too much water resistant.
Okay.
Yeah, so what we can do for its coating, in the sense of coating, or how we can make it as a hydrophobic material, so it will become as a water resistant.
Mm, it is very tough for me to give the answer,
Oh, okay.
uh,
Sure, sure.
because material, those who are, how can I say, materials, those who are not hydrophobic, you have to apply like silicon based material into the
Okay.
finishing. That then maybe you will improve the situation. That maybe
Ma'am, is any idea
silicon based.
regarding natural rubber latex?
No, I don't have.
Okay, okay, okay. So I am planning to take trial on it.
Okay.
Okay,
In that
okay,
case, best of luck to
okay,
improve
okay.
the situation.
Yeah, okay. Thank you, ma'am. Thank you.
Welcome.
Thank you also, Sangram, for your showing the example of the bio-based ladder.
Bio-based material, yes.
For now, we got another question in the chat box. I will read it for you, Dr. Zohra.
Yes.
From Bayu, what is the treatment strategic executions aim making the high quality of skin or height from regional agroecosystem which resulted by climate and weather accurate? So I think this is the
This is very tough question as you know, it is natural. So due to the climate, due to the environment, due to the food habit, naturally we will able to collect from the nature. I mean, animal hairs and skin, isn't it? It's very natural.
yeah, maybe there's some response from Bayu. Is it clear or do you
Yeah,
want to ask the question?
thank you. Assalamualaikum warahmatullahi wabarakatuh and good morning. My name is Ahmad Bayu Aryawan, the alumna of Animal Science Faculty in Haru Oleo University in Kendari, Southeast Sulawesi of Indonesia. I would ask about uh, the regional ecosystem. For example, body cow, and peek lamb that uh, that live in tropical in a variety tropical regional, ma'am.
Yeah.
Like cold or hot region. I think what's the treatment strategy execution? I mean, what's the what's the um, how to uh, the strategy come for resulted skin or height, high quality of skin or height with climate and weather accurate daily in every day, madam. I mean, thank you.
Okay. Thank you. So basically, I focused on leather processing, quality control, productivity improvement. So I don't have that much knowledge regarding animal husbandry or that section. So still, I can say for climate change or day-to-day care, taking care and food, these sorts of activities and nutrition, these sorts of activities may helpful to uh, support the animal growth uh, to for better for quality hides and skin to develop quality hides and skin of the animal. So for the better hides and skin, we have to provide food, proper food, nutrition, environment as well. We have to taking care of every day so that we can collect better hides and skin from the animal.
How is it
Yes,
for
Abhishek.
Bayu? Is it clear now? Or do you need to ask more questions?
Yes.
Yeah.
Okay.
Thank you, Dr. Zohra, for your answer. So I think it's clear for Bayu. And now I think we don't have any more questions. So before you leave, we're gonna giving the certificate for Dr. Zohra, but because you're on the Zoom, we're gonna share screen the certificate and we're gonna take a photo of it.
So I'm stopping my slide here.
Yes, please.
Yes.
Yes, we're gonna share the certificate shortly.
Okay, thank you so much for the certificate.
Yes. Okay. Our team gonna take the documentations. Okay, we already took the photo. Thank you once again, Dr. Zohra. Insightful material. It's gonna help us for the understanding general code.
I am really delighted to be a part of this program, and thank you, organizer. Really, it means a lot. Thank you so much.
Thank you so much, Dr. Zohra. Hope we meet again soon.
Okay, thank you. So I'm leaving now. Okay.
Yes, you're free to go now if you want to go.
Yes. Okay.
Yes, okay. Okay. So now before we go into the next sessions, I want to remind you that for the offline participants, you're free to grab a drink or grab a coffee or even grab a snack or go to the toilet in the sessions. Just make yourself comfortable and don't being tense up. Just relax and everything. And also for the online participant, I remind you again, please, I will be much appreciated if you turn on your camera and put on your, maybe for the
Apa
audio
ini apa ini?
participant, because we have our second speaker now in
Sama
the
juga dengan
Zoom.
ini Unhas.
So Ravindran, how are you today?
Cuma dia menyusul.
I'm fine. And you all?
Bapak menyusul
Yeah,
acaranya
I'm good too.
mulai
It's nice
dari
meeting you
hari
again.
kemarin juga.
It's been a long time since we met the last time.
Ada nanti prakteknya ini, ya, ada
So
prakteknya.
now,
Ya,
for
ada
all the
prakteknya nanti nih,
participants, I'm
tapi
gonna
dia
introduce
offline.
our second
Dia hanya
speaker.
untuk offline.
So,
Ini kan
Dr. Bala
online
Subramani
teori
Ravindran,
toh.
or we could call Dr. Ravindran, is currently serving as an associate professor in the Department of Civil
Mana ini
and Energy System Engineering in South Korea. He is an active researcher whose work focuses on solid waste management and
Tentang apa ini
wastewater
Bapak?
treatment.
Letter juga. Leader juga.
His current research include biocar application for reducing greenhouse gas emissions
Munir,
during
ya
livestock
Allah.
waste composting, as well as aerobic and anaerobic
Dapat jadi juga,
digestion of various
iya sama
domestic
dengan yang
and
ini.
industrial wastes. And for today,
Betul,
Dr.
betul.
Ravindran will bring the topic cleaner weather productions, trends, and alternatives to conventional. So please, Dr. Ravindran, time is yours.
So, good morning, everyone. So that thank you, Dr. Zainal and other members who get into me for this wonderful international series seminar. So today I'm going to talk my topic on that cleaner leather protection trends and alternative to conventional. Actually, in this topic,
Lining sekarang
I'm
toh.
going to split
Saat ini
into
masih dalam
two
lining toh.
ways. So one is like a general about
People
cleaner
sebelumnya
trends and
ammonium
alternatives to
sulfate.
conventional. The second one is alternative trends for
Natrium
leather production
disulfida, enzyme
techniques.
binding
And
agent.
third is about that during leather production we can get so many byproducts, so that we
Ya,
are
Prof.
going to also see that utilization of
Melarutkan protein dan
leather
kolagen.
products through different processes. So now we're going to start about that cleaner leather production trends and alternative to
Kulit yang
conventional.
fresh.
So what is the cleaner leather production industry? This is sustainable leather manufacturing with reduced pollution, water use, and toxic chemicals. Also, the focus is on eco-friendly tanning, waste minimization, and resource efficiency. It also should support circular economy and the green manufacturing process. However, the environmental problems in conventional leather processing, because of the chrome tanning dominates during that tanning process. We use in the chrome process chrome. So the chrome tanning dominates about 85 to 90% of global leather production. It generates large quantity of toxic wastewater, chromium containing sludge, high COD, the chemical absorption demand, and the biological absorption BOD effluents. It's also the solid waste from hides and the chemicals. It's the high water and the energy consumption. It's during the process. It's require high water, huge water need. Also, much huge energy consumption process. So also the key drivers of cleaner production. Strict environmental regulations needed
Udah.
like REACH and EZDHCC. Consumer demand for sustainable products. Industrial need for low carbon manufacturing process and also ESG and circular economic initiatives. What are the current trends in cleaner leather production? Is that chrome
Banyak
free
papain.
and metal
Papain,
free tanning?
bromeliel.
Because of that in that conventional process they always using the chrome. So we now that researchers are trying
Nah,
to
kalau buat
without
sendiri
chrome also
bagaimana?
without
Itu
metal
yang biasa
process.
dipakai untuk
It's
marinasi.
look like using of the vegetable tanning from bark, seeds and plants. Also they using this zeology based tanning process like zeology like this way. Now altogether free and heavy metal free systems. They also used enzymatic and biotechnical approaches
Waduh.
like enzyme assisted dehairing and batting, reduced sulfide and lime usage in the process. Also, it's become lower waste also toxicity. On another way, water and the energy saving technologies. Water is very important these days, so we cannot use much water for any process. So that in this process we can use the lost salt curing, water less or reduced water tanning processes, recycling of processed water, also energy efficient drum systems.
Rasa jarak.
After the waste, example, after they use this process, we also produce so many
Hewan.
waste. So waste valorization and circular
Dari
economy is very
ini,
important
bro. Dari
steps.
mana ini?
So in this process, a recovery of collagen and the gelatin
Dari
from the leather is
bukannya
one of
ini,
the main
dari
products.
apa itu yang
Also conversion of
lumen?
dead sludge into
Bukan.
bioenergy or biofertilizer. After waste, we can be that waste. That after that the wastewater, the tanning process, something they also produce the generator
Iya.
sludge. The sludge we can be
Iya.
converted into bioenergy or biofertilizer. So the reuse of trimming and the flushing waste, the flushing waste. Example, the hide, it means like a skin. The skin beneath layer called flushing. The flushing is not like a full flesh. It's like it called like a adipose tissue. It contains so many proteins and lipids. So how we can use this uh, resource for some other products. So I also done my PhD in this area only. So we also going to be see on my third part of presentation. So also the digital and the smart
Kira ada.
manufacturing, AI based process, AI based process monitoring, automated chemical dosing, cleaner production optimization. It's also reduce the like energy consumption, also that cost effective process like that. So what are the alternatives to conventional leather? So even though because of we using the so much animals for leather production, even though our aim is that faculty of animal science, but still we need to minimize that usage of animal, because so that we can be, because of that if the production rate animal production rate is low means we cannot use much, so we need to balance all this. So we can be also used as a bio based leather alternatives, such as plant based leather, like from the pineapple leaf fiber, like they call Panatex, also cactus leather, apple waste leather, cork leather. Also from fungi like mycelium leather also available, produced from fungal mycelium, biodegradable and renewable process, and a lower carbon footprint. If you using like this types of leather, so it's very easily biodegradable. Also, we can use that renewable process, also lower carbon footprint. If you're using that a chemical tanning process or like animal skin like that, it make more non biodegradable like that way. So also the lab grown leather. In the lab, we can also grow leather, cell culture leather without animal slaughter. Also emerging biotechnological innovation. So synthetic alternative. What is the synthetic alternatives? If example like a polyurethane leather, PVC leather, microfiber leather. That apart from that animal leather, we have so many leather options is there, like a plant-based leather, fungi like mushroom leather, lab-grown leather, also synthetic alternatives. Also, what are the advantages? Is reduced animal independence, also lower toxic emissions, improved sustainability potential. What are the challenges still in this process? Durability limitations, high protection cost because of if we follow some other process for minimize the other things means we need to spend a lot of money. So still some challenges there in this process. So also that some still contain plastics or PVC coating. No plastics. Now we also worry about plastics. So, what are the future perspectives and conclusion? Future perspectives means like a fully biodegradable leather materials we are focusing on. Also, the green chemistry in tanning process. Also, nanotechnology based finishing process. It should be carbon neutral leather protection. The industrial symbiosis and the zero waste transition should be very, very important. So, what are the key sustainability goals? Example, we should minimize the chromium pollution, also the reduced usage of water, also the energy consumption process, improve worker safety, also enhance the biodegradability and the recyclability. So through this process, what we need to take home, like a conclusion is, cleaner leather production is essential for sustainable industry development. So chrome-free and bio-based technologies are rapidly growing. You know that because of combination of biotechnology, green chemistry, and circular economy approaches will shape the future leather industry. It should be because otherwise, in India, in before 10 years, that leather industry and the economy is very huge. But these days, because of the pollution, they're not able to control much. So that slowly decline. So still we are working on that. But however, when I was in Indonesia, it's all good. They're doing some good. Okay, what is the take home message? Further leather production must balance performance, environmental protection, and also sustainability process. Okay, as before I talk about it, so this is my second part. So the same thing, like a sustainable alternative to conventional leather protection. So why alternatives are needed? Because of conventional, the same thing, but still we're going to talk some more like that. No, same thing, you know that plant-based leather, mycelium, lab-grown, the advantages. Also, synthetic. We used to for synthetic. So what are the challenges? Like a high production cost, limited rod production, comes like that. So same thing, but only what we need to understand, apart from that current leather production process or the current leather using the animal, we can also use it as a plant based or like a fungi based also synthetic waste. So only what we need to do, we need to optimize, we need to reduce the cost for that production. Also, we need to reduce the energy consumption process if we're using that new type of leather. This is we should be do for thinner production. So this is my last topic. It's like very interesting topic for me. You know that I told you about that. This I'm going to use that proteinous solid waste. It is solid waste generated from the hide. You know, hide means skin. The skin we cannot use to directly the skin converted into leather. So we need to scrap that hair, we need to scrap that some skin adipose tissue, then only we can use it for some leather production process. This work I all used to from pre-tanning process. You know the pre-tanning means before tanning. Before tanning process means like a chromium, whatever they use in that. So post-tanning, pre-tanning is there. So these all are used from that pre-tanning process. I took some topic on studies on the fermentation. I used this fermentation technology, two different fermentation technology, solid state and also submerged fermentation of protein solid waste. Also some other split, bacterial and vermicomposting of fermented solid waste. Also finally, application of biofertilizer into the commercial crops. You know that is like uh, a zero waste technology. If we're getting any the tangible solid waste as a byproduct in pre-tanning process, we can be first produce enzyme through the fermentation process because it's containing so many proteins, the waste containing. Further, we can also use that remaining undegradable waste. I used the process called bacterial vermicompost. In this process, we can be able to produce that biofertilizer. Also, the final application of biofertilizer commercial crops, like so many crops is there now, we the daily used crops. So in this process, I am not using that bacterial and vermicompost water, I used directly that hydrolysis, fermented hydrolysis, like as a liquid fertilizer. So we're going to see about that now. So tannery solid waste. What are the tannery solid waste for this in this area means animal flushing from leather industry. More than 10,000 tanneries are spread across the globe with the annual processing capacity of 9 to 10 per 6 tons of hides and skins. India has about we talk about India because of I done this work in India, so we have the 3,000 tons in the total processing capacity of 700,000 tons of hides and skin per year. So major concern India. A different area. You know that like here Jakarta, Bandung, Samra is there like that in so that Tamil Nadu, West Bengal, UP, Punjab is the so many states are there. So we also produce so many tanneries like the leather things. So out of the generation of solid waste, 1 metric ton of wet salted hides. You know that after remove the skin from the slaughtered animal. So we cannot just start the process because of that slaughterhouse. Uh, we cannot do the same process in that area. So we need to preserve, and then we need to take all the hide or skins to
Prof
that
Irsa, Prof Irsa,
protection
minta
area.
maaf sebelumnya ada
So
Pak Bu Alimnya nih
until
di kampus.
we need to preserve means we use the salt
Di ruangannya di
like
mana
that.
beliau?
Along with this 60 kg of tanned and non-tanned solid waste and 100 kg of raw material is lost. You know that when 1 ton means 1,000 kilogram, only 300 kg only it will become leather product. Remaining 700 kg almost it will become waste. So these
Industri
solid
pakan.
waste are distributed as
Di mana
animal
itu Prof?
finishing, chrome shavings, chrome
Oh,
spits,
dekat
and buffing
Ternakpos.
dust and skin clipping and hair. It's all I'm not talking about the pre-tanning process or post-tanning process. It's all cold tanning process here. So
Oh,
among the
di
solid
lab
waste
ini,
generated by the
lab
industry, animal
industri
fleshing
pakan.
draws attention of innovative scientists and technologies for high recovery of value-added products. Now that I'm not
Oh,
talking about
di
the
samping tanam
fleshing is
kan
not about
potong
not
yang
to eating.
kambing
It's like but
dan
from the
kambing.
skin. You know that you can see this man who
Oh,
scrap that
di belakang.
some product from that hide skin is called animal fleshing.
Ya,
It's called
yang di
also
NBC itu.
adipose tissue.
Bukan.
This contains so much of proteins and lipids. So we need to replace. Currently the
Kiri.
so many processes are placed as a
Oh iya.
animal feed production or something, but still due to is a huge amount of protein and lipids, why we can't produce the protein enzymes and the lipid enzyme. So this area is like that. Okay.
Bayu
So
ingin beli
exporters and
bukunya
service
Pak Alim yang The
currently
Engine
how they
The
used
Poultry Engine.
to dispose this waste using the conventional methods like a land court disposal,
Ya,
anaerobic digest process, pyrolysis, and incineration. Also, they produce do manufacture also
udah
animal
karena
feed production.
kalau misalkan
So
jauh
what
nggak
we can
apa-apa,
do,
tapi
what we
kalau
can
yang
do,
ini,
like hydrolysis of animal fleshing through solid state
tapi
and
ada
preservation
kontaknya
stability
Prof.
process. Also, we can produce
Iya
some
Prof,
benzene.
nanti baik
Also,
catat
next
dulu.
step is my areas like composting, vermicomposting, terrestrial solid waste using short reduction period. Why short reduction period? It means normally the composting and the earthworm using like a vermicompost process need lot of time like 100 days. So how we
Mohon izin
can
Prof.
reduce the days time of producing the biofertilizer through this process. So this is the area. Also fermented fertilizer for commercial purpose. The fermenter now after fermentation, this contains so much of nitrogen, phosphorus, so much of nutrients. So it can be used as a liquid fertilizer for commercial crops directly. So this is flow chart. No, whoever doing PhD or master students, so please making like the flow chart before you starting your uh, thesis work. So it will be helpful for you all where you want to improve, where you want to do more work, what you need, what you don't have, what you have, who you need to approach. So it's all able to help for this kind of flow chart. So in my process, I used to characterize of animal fleshing, also isolation of like this, syndesmus, also clostridium, water ferment animal fleshing. Why I need to isolate means I need to find or isolate
417474.
particular bacteria
for
28 berapa?
this fermentation produce to
27.
produce
Oh,
enzymes inactively. So I isolated these microbes.
087.
Also, I used two different processes. Why two different processes? Means, example, submerged fermentation means we need a lot of
Oh
water,
ya, 0812
lot of
417.
medium solid state fermentation means we need just 5% of water is enough. So we use just the solid state.
4087.
During the solid state also we are able to produce enzymes. This is called solid state
0812
fermentation process. In this process, in
4174.
this two different process, I used two different microbes. Why I brought two
087.
different
Baik, Prof.
microbes?
Terima
As
kasih
I said,
banyak, Prof.
around 100 microbes
Yes.
out of 100, I used these two type of microbes only able to produce
0812
high
4174
quality
087, Prof ya.
or high volume of enzymes like that. So I said that. So what are the enzyme produced means like a protease. So we cannot use just raw protease. We also the protease protection, purification, characterization. So I also checked which is the best performance. So these are the best performance. And then later the ferment annual flushing just I applicable into the battery composting process or vermicompost process for the production of biofertilizer. Also application of biofertilizer into seed germination process on the various crops. And on other hand, here example, this direct liquid fertilizer I used to for to comes tops called tomato. So tomato crops for the evolution of yield potential, how much yield compared with the control you
Sorry
to
to
reduce
interrupt
the
you,
stuff discussion.
Dr. Hindran. I
Yes,
think
yes.
your screen is gone. Can you share screen again? Or is
Oh,
it?
yeah. Oh my God. Okay, just wait.
Okay.
Is it okay now?
Yes, it is okay now.
Just wait. I also want to check here again. Just wait.
Tinggal 2 menit lagi. Terus lanjut training.
Hear me okay now?
Check, check.
Hello?
Yes, it is okay now. It is very clear.
Okay, okay. Thank you. So in this work is my overall work. Also, it was my PhD work. So I used to this waste called animal fleshing, or we call this adipose tissue. That waste. So it's my PhD topic. So that overall, what I explained before. So we should also check that alternative leather production process. Also, we need to focus on environmental friendly, toxic free, cost effective process to do this. So also so many alternatives are available like plant based leather or something, but still we need to optimize that cost, also that duration process like this. So it is my previous process. So in this area, this part I focused on leather byproducts, especially pre-tanning process, before tanning process, because of after that when using that hide or skin for produce the leather. So we need to scrap to remove the adipose tissue. It is available that beneath of that skin, like a hide, like we want the any animal, like what we using for the leather production. So that fleshing contains more
proteins and lipids. So we in this process, I also able to produce enzyme through the two different fermentase process, like a solid state fermentase process and submerged fermentase process. Also that that hydrolysis to animal fleshing also applicable into two different process called bacterial and vermicompost process. Through this process, I will be able to produce biofertilizer for that some crops. Also, directly I applicable without compost or vermicompost process. Directly, I was trying to utilize as a that hydrolysis as a liquid fertilizer for some commercial process, especially like tomato. This is my purpose. So how I isolated? Like example that when isolated, I seen like a gram positive microorganisms, also the fermented type 1, also trypsin. Also when I analysis like cystinase or DNA sequencing data, it's indicated that Clostridium limosum with 80% similarities to those of nearest strain Clostridium. So what are the source is actually I want to tell where I isolated this source. This microbes means I isolated that excreta of flesh-eating animals. Flesh-eating animals in the zoo. I was doing my PhD. I was done my PhD in India, and then my institute opposite one zoo was there. And then I visited, and then I got some special permission. I take that excreta of flesh-eating animal, and then I take that, and then I used some
Aduh ya Allah.
other culture process. After so many process, I isolate these microbes because of is easily because of we also use in the flesh like that way, but we don't need to use always like this way because of even though if I isolate from soil also
Aduh.
that microbes also you able to
Tangan ini kan
degrade that piece of
kiki.
waste. Because of example, if you go to any leather industry, so if they throw somewhere like hide or something in the
Biarlah
in that
begini aja.
some
Daripada
land code is
ini ya ini.
if you go there,
Ini ada ini
if
ya.
you take the soil, you can isolate use like the same kind of
Deh.
bacteria.
Kayak
I
film
used
Korea
to like
ini.
this way. So even though we are doing any kind of work, we need to analyze the characteristics raw material, what we used to. So that what I analyzed this raw material. So it's a composition of carbon, hydrogen, nitrogen, and sulfur. It's around 36% of carbon, and the hydrogen 18, and the nitrogen 11. So it is the perfect for used as a fertilizer. However, we cannot use it directly to just flush into plant for that's what it is. So we need hydrolysis. We need to degrade it, and then we need to. It should be easily available sources for the plant. So also it contains that what are the chemical compositions like total organic carbon, total nitrogen, total gender nitrogen, and the CN is 3.7 total solutes, ash content water is like that way, and also that minerals also available. It's like iron is 30%, 30 milligram per kg, and also manganese. It's like that 18 milligram per kg. So we used two different fermentation process using that two different microbes called Clostridium limosum and Cellomus ruminantum. So this process, what I understand, and the fermentation process is just the microbes cause an organism to break down into simple substances from the complex substance using the action of enzymes. You know that when that microbes used to utilize that way, it should be produce enzyme. That enzyme. So we can that enzyme what we understand we can call it protease. So two different processes. It's called a liquid sub-based fermentation. It's called a liquid state fermentation. Solid state fermentation is called like without water or absence of near water. So we also we can't just use that fix that time or medium pH or equilibrium temperature. We need to optimize. We need to optimize even any process or any work. We need to optimize. We need to optimize. So we also done that like a effect of fermentation period like 24 hours, 48 hours, 72 hours, 96 hours, 120 hours. Also effect of pH like from 3 to 8. Also incubation temperature. We don't know the temperature that the enzyme or that things liable to that room temperature or 40 or 50 or 60. It's like a thermostable enzyme or thermostable bacteria. We don't know anything. because of still what we understand only, okay, the enzyme able to produce only, but through this process only we understand, okay, how it's like that. Also we cannot use to, because we have still that all the waste, but still we need to use to, like slowly, like 0.5 gram or 1 gram, 1.5 gram or 2 gram only, like that. So in that fermentation process
Apa
we
fungsi
used
pendimangan tadi?
four different important
Apa fungsi
uh,
fungsinya?
chemicals. So these chemicals able to
Ito.
immediately give the source for the
Ah,
bacteria
wait,
growth.
wait.
Later only in this
Biru.
source only the microbes are able to
Ada.
survive,
Masih
and
ada
then
tuh.
only they started to degrade the
Biar
fleshings.
Bayu yang ambil
So
juga.
we initially we need to work ammonium chloride or
Biar Bayu
potassium
mau.
hydrogen phosphate, and then potassium hydrogen dihydrogen phosphate, sodium chloride.
Sudah ada ini.
So after this kind of experiment, we got some data. We used some method to identify called response
Ini anak minta
sulfate fermentation
dulu.
process. In this process, we able to easily identify which temperature, which incubation period, what are incubation period, or which pH, how much gram we need to use for that kind of particular enzyme. Is easily able to identify. So we identify that. Then so these are your submerged fermentation for the process limosome, submerged fermentation monas. So also the solid state fermentation, fermentation for this two, but it means like a two different fermentation process for two different bacteria. So what we understand, we used in the submerged fermentation process. It means called liquid fermentation process. Maximum activity we used, we can get, we got from Selenium monas ruminantum. Also in salsa fermentation process, also the Selenium only shown huge result. So up to temperature is 72 hours, and the temperature for that process is 40 degrees Celsius, and the medium pH is 6, and the mass of 1 gram is enough. Because of we used to for 0.5 gram, 1 gram, 1.5 gram, 2 gram like that. So what we understand, this is optimization for that process. So after that, we are also able to produce enzyme. What are the how we need to purify that extracted enzymes? It's called like the ammonium sulfate precipitation process. It's dialysis. We used to for the also cellulose column chromatography, also the Sepatex G under chromatography for the two different process. So we also identify that through submerged fermentation process, salt fermentation process. Here also we able to produce the, pure or higher molecular weight from Selenonas or ruminantum producing bacteria producing enzymes like protease. So it's all like the same thing. Like what are the basic chemical processes for the purified acid process, like pH, thermostable inhibitors, metal ions, surfactants, solvents. We used just it's also able to characterize the purified protease. We identify that enzyme is acidic enzymes. So we also analyze that some we also analyzed the scanning with the microscopy. That after hydrolysis process, still some debris is there. The debris we checked how it's like a 0 degree and the final degree. How it's any different. So easily identify that degradation. You can see this through the submerged fermentation process. Because of why we used only for choosing the Cellomonas, because of we identify this bacteria produce stable enzyme, also that it's like they're producing more enzyme, are able to easily purified. So based upon that, we focus on that. So we focused on that flushing, how it's degraded. So we easily identify. So if we compare initial both fermentation process, submerged, like a liquid fermentation process or solid fermentation process, this also completely or maybe mostly hydrolysis. So, what are the interpretation? Submerged for more rigidity of fiber networks and hydrolysis sample compared to less rigid nature, detach- de- detachment of protein fibers in the hydrolysis sample present in moderation of animal fleshy morphology under submerged fermentation process. In the solid state fermentation process, the unhydrolyzed animal fleshing showed fibril networks with a rough membrane structure and fine distribution of clustered tissue fibers. They also we identified that after hydrolysis, shown some distinct spaces occupied by the liquids and hydrolyzed peptides. So, moreover, the proteins organization along with the fiber result in microstructural changes in the fleshing. This also confirmed. Okay, you can see this here. Hydrolysis. How you can see this. This is
Sama.
liquid fermentation process like submerged also solid hydrolysis. You can see that hydrolysis how it's happening. So what we
Sama.
understand through this process, animal
Sama.
flexion completed hydrolysis into solid state fermentation when compared submerged or liquid fermentation process. In between two isolated microorganisms, Cellomonas renomentum shows a better hydrolytic activity. So after this process, I told about the three different processes I done like enzyme protection and purification through this fermentation process, also the bacterial
1000.
combustion and vermicombustion process, also that
1000.
application of direct distillate into some composting crops. So we used to now that we're going for bacterial composting process. So the composting, what is the composting? Is the recognized as cost-effective method for solid waste management. So for the conversion of manure, only small portion of the total organic carbon and nitrogen were released by microbes, but during the composting process and the complex were left unmetabolized. So we normally so many composting process is also able to produce that fertilizer, but still only small portions or maybe undegraded materials are still there. So in this regard, we used bacterial composting, like a bacterial implement like that. So person work involved utilization of hydrolyzed waste obtained from the solid state fermentation and submerged fermentation using isolated bacteria, Pseudomonas. This hydrolytic peat treatment hormone aim to accelerate stabilization and facilitate transition into complex into simplest and ultimately reduce the composting period. If example that flushing we directly if we want to do produce biotracer through the compost process, it will take a lot of time. You know that the example we just put 1 gram in the soil, it takes a lot of time to become biofertilizer. But if hydrolysis through the fermentation process, then if you do this process like bacterial composting, it's able to easily is that time compost time much be reduced. So this is one big advantage. So we cannot just do the composting process with the hydrolyzed waste. We need some cow dung and also leaf litter. I don't know the scene ratio. The scene ratio just a 3 only. So we need to adjust. We need more active. So cow dung contain more carbon nitrogen. Also so much microbes is there. The benefit that microbes also helpful to degrade further undegradable unhydrolyzed flushing in this process. So along with that, we also used leaf litter. So we adjust the seed ratio. When starting the seed ratio is very important for the producing the biofertilizer. So we used for cow dung and the leaf litter. So these are the experiment design, like control ones are submerged fermentation liquids, solid fermentation liquid, using that both for cow dung and also that things. 1 liter this. Then what we understand here. Okay, these are the analysis we used for specific chemical like pH, nitrogen, phosphorus, potassium, heavy metals, microbiological analysis, enzyme analysis, instrumentation analysis called FTR, Fourier transform infrared process. Also TGDSA. What is it is called like stability of the product. Be able to identify the stability of product. So these are the parameters we studied for this kind of things. So what we understand. Okay, I can show here. Yes, we have within this small time. Okay. So you can see that during this process, that pH initially it was around 77.3. After the 28 of the composting process, it's become slight acidic only, not like alkaline. So it is good indicator for that it should become biofertilizer.
Screen
Also
share
that
that
easy.
has been sent.
Yes,
Yes.
sure.
Already you have 5 minutes remaining for your
Oh,
time.
really? Okay. Now I can go back here. Okay, so through this process, what I understand in the end of battery composition process, the complex substance were mineralized, the nutrient value was increased in all treatments, especially solid state fermentation process were better compared to all treatments. However, the nutrient value is able to increase further when we composting using earthworm. Earthworm, you know all about that earthworm process. So that even though battery composting process were I did, but still we need to improve the quality of biofertilizer. We also did earthworm
Air.
process called vermicomposting. So in this process, we used to Hebrews egina is one kind of earthworm. Is from African type of earthworm. So this is the earthworm. You can see this during the vermicompost process. The physiochemical, microbiological, enzymatic demonstrations were followed in the same bacterial compost process. That we also checked that through this process, what we understand. Um, what we understand, it's become 5 times more nutritional value than the product of battery composting process. So we want to check that how it's using that germination index. So we also using that different kind of seeds like tomato, cucumber seed, like bottle cut seed, also green gum seeds. So using the 4 different commercial crop seeds we use for the check that we identify that there is no toxicity. So why we need to do this kind of the germinal synthesis on the lab level means before applicable in the land like agriculture land. So we need to just check in the lab level. So whether is that manure has any toxicity by toxicity or not. What we understand is very good because of how we analyze it means if you put 10 seeds means if it grow more than 5 or 6 seeds in the better plate means it's very good. More than 8%, 8 number of seeds is grown means it's very good. It's called it's one type of in maturity
Prof,
in
jadi
maturity process.
yang lebih berat
So we
itu
also then we go for
apa
this
lebih berat
kind of things.
kalau air dan ininya
We
jenis
also
jenisnya
use
sama
the
ya?
directly biofertilizer from
Maksudnya
directly
yang disampaikan tadi
use
apa
the
bedanya? Apakah apa
hydrolyzed
jenis apa
freshly
berjenis air
from the
kemudian
two
itu
different fermentation
apa
process
sama
is
dengan
also
satu
called the
liter dengan
liquid
satu
fertilizer.
minyak
We
itu
also
kok?
applied for the tomato plant in the pot culture method. So we also identify that. So here, although the solid state fermentation liquid fertilizer can produce more yield than submerged liquid fertilizer. So you can see that the tomato also identify that. We also check that compare with the NPK. What is the 25%, 50%, 70%, 100% means we are not able to directly just spray from the directly the fertilizer. We also diluted like first 25%, 50% to 400%. Also we compare with the control also NPK. It was very good only. So, what are the summary and conclusions? This flushing, animal flushing is a major transition of waste during the manufacture. It should be disposed environmental friendly. Also, this record we focused for 2 different
Berarti
bacteria
berjenisnya
for
sama, berarti
hydrolysis,
pengemulsinya ya
two
di dalam
different
pengemulsinya itu.
fermentation process. After that, we also do done for bacterial vermicomposting process, and along with it, like used as for liquid fertilizer for tomato plant. So what we understand, this study provides the value-added products at the end. So it could be we can use it as a biofertilizer, liquid fertilizer with this waste. Okay, thank you for this attention.
Okay.
So, but please, do you have
Thank
any
you for
questions,
your
please?
yes. Thank you for your interesting presentation, and it is so insightful that you were talking about the cleaner alternative for production of leather, right? And also for the treatment for composting. And for now, for the participants, do you have any questions for the offline participant or online participant? Please raise your hand.
Garam.
Okay, then while we wait, I think I'm gonna ask you questions. So, in the earlier for your presentations, you were talking
Dikasih
about
kasih
the
kulit
bio-based
buat
leather
gede-gede.
alternatives such as lab-grown leather and also the mycelium, the mushroom leather. Could it be produced in a high quantity, or could it be used such as the same way for
Dede.
the conventional leather to make maybe like a jacket, like a wallet,
Oh,
or like a shoes?
konten.
Yeah, what I understand actually, that still it's like that. It's just alternatively we
Sepertinya
need to
tidak bisa
optimize that
ya.
production cost or like that source requirement. So, but in the lab level or like some plan, we can uh, produce that fungal kind of leather in that some form level, but still we cannot use to like we cannot produce like a big coat something like that. So we can just what you understand what we suggest is like a wallet or belt or something we can use for them. But for some other things like synthetic leather or some other leather can produce also like big
Rumah
size
potong hewan Samata
product.
yang ke arah jembatan kembar.
So we need to check that how it's cost compare that
Iya, sama ngapa?
conventional how is sources availability. So we need to focus on that reduce the
Betul.
cost also that energy consumption in that alternative
Kenapa, Prof?
products.
Mau ke...
So we can be successive for this alternation.
Okay, thank you very much for your answer. Also, for other participants, maybe we could take one more questions, online or offline.
Okay, thank you. But
Assalamualaikum, sir.
yeah, sure.
How to get PDF copies of all lectures?
Okay, so please
Iya. Iya, Dek.
you can answer.
How to get PDF copies of all lectures?
Okay, got it, Mr. Mamun. So the committee will compile all the PPT files from the all lecturers, and maybe in the end of the session we could give to all the participants for your presentations. Is it clear?
Thank you,
So,
sir.
if example, if anyone want to contact me, have any doubt or any help or any things about the career, any studies or anything, so please contact me. Uh, Email ID kalamravi@gmail.com. I already mentioned in the chat. So please write with me if you need anything. Things.
Okay, thank you for your time, Dr. Ravindran. So if any participants would like to contact Dr. Ravindran after the sessions, you could hit him up in the email in the chat box. He already sent it in the chat box.
Thank you very much for. Okay, thank you once again. Bye bye.
Hey, wait, don't leave now because after
sure.
we're gonna giving out the certificate for the sessions. So our team will share the certificate through our screen.
Oh, sure.
And yes, we will take the documentation first. So wait a minute.
Okay, thank you for this. I would like to also share one thing to that audience. I think it may be that undergraduate, postgraduate, PhD also some lectures there in this
Bali
talk.
tidak ada.
So if anyone want to write paper or do you want to know how to write paper in easy way to publish in a very good journal, please you may contact my mail ID. Because of I should suggest please at least you should publish one paper during your bachelor's, master's or PhD something. So for masters and PhD students, please publish in Scopus high quality Q1 journal. For bachelor's, still you can try your best. Yes, you have only short time, so try at least one paper published in any national or international journals. In this regard, anyone need any guidance along with your current supervisor, I able to give my suggestions to you all.
Okay, got it. Thank you for the insights for Dr. Ravindran. So for all the participants, as you hear from Dr. Ravindran, we will just ask about the maybe like suggestions and everything for the undergraduate and also the master degree, because Dr. Ravindran is also notable researchers, and you can hit him up. Just find you can make yourself just contact him by your own. Okay, thank you once again, Dr. Ravindran, for your time
Thank you.
and for your
Maaf, maaf,
insightful
maaf, maaf.
materials.
Thank you.
Maaf, maaf.
Yes. Have a good
Air
day.
sudah.
Bye bye.
Toh,
Air sama garam.
garam. Baru kasih masuk asam formik baru.
Okay, so now before we moving on into the third speaker, I see